Great. So thank you for all of you for attending, and thank you, Dr. Riza, for giving a great presentations. My name is Leo Chan, and I'm a science and technology advisor from 10x Genomics. So today I would like to talk about how you can use our 10x Chromium single-cell gene expression solutions to resolve complex biological systems. So as Dr. Riza has mentioned, the biological system is very complex. So for examples, if you are cataloging the trillions of cells in our body, or if you're detecting millions of variants among the billions of bases in the genomes, all these require complex and modern tools for you to assay them. So Dr. Riza gave a great examples on the study of infectious disease. Now, to change the subject a little bit, if you're conducting cancer research, for examples, in addition to look at the genomes of the tumor, it is also very important for you to look at and characterize all the cells that are present in the tumor microenvironments. So to understand biological complexity, single-cell analysis has become an indispensable tool. But then why exactly do we want to do single-cell biology? So single-cell genomics and transcriptomics can reveal biology in a much higher definitions. This is a powerful techniques for you to resolve cellular heterogeneity. You can use it to identify rare cell populations in your samples. You can compare the transcriptomes of individual cell populations. So this allows you to characterize complex tissue environments. You can look at the characteristic of gene expressions, and you can use the data to follow and trace developmental and cell lineages among many other applications. So for the single-cell analysis, the 10x Genomics Chromium systems enables you to profile the transcriptomes and the epigenomes from the single cell. So today, I'm going to focus on the transcriptome side of the story. But why do you want to study the transcriptome? We know that the transcriptome, that is the gene expressions, this is the key determinant of the phenotype of a cell. And it actually controls the identity and fate of all the individual cells in your samples. But one of the issues that we have is much of what we know about the structures and the functions of the transcriptomes come from studies averaging the measurements over a large populations of cells from your samples. But we know that in these samples, it is functionally heterogeneous that composed of many different cell types. And therefore, traditional bulk RNA-seq techniques or microarray studies, they actually hide this variability between the cells in your samples. Because in bulk studies, you're looking at gene expressions across many individual cells. Therefore, the only readout that you get from bulk RNA-seq data or microarray data is a single point readout that represents the combinations or the average of many of the cells present in your samples. One way to say this is bulk data prevent us from looking at the nature of heterogeneity in the cell as a molecular level, because that is a basis for us to understand biological complexity. Now, in the single cell studies, however, it allows us to tap into cell-to-cell differences. And we know that these differences in any tissues or cell cultures are an essential features of the biological state and the function of the single cells. And you can only tap into all these informations by doing single cell studies. Showing on the bottom of this slide is a two-dimensional t-SNE plot that you can look at the high-dimensional gene expression data. So each of these dots is a single cell. You can cluster the data based on gene expressions informations. So for example, cells in one cluster in this diagram is color-coded, and they are characterized by the same known cell markers. So the precise characterizations of a heterogeneous and complex biological samples can only be achieved with the single cell studies. Now, single cell profilings allow us to compare specific subpopulations of cells. Therefore, single cell analysis is very sensitive and is more precise compared to bulk RNA-seq. For example, on this slide, you can see that we can easily select and identify these myeloid populations between these AML or acute myeloid leukemia samples to compare with the normal PBMC samples without worrying that the signals from this specific population is going to dilute out by other populations. Now, the bottom of this slide shows the overlaps between the significant gene sets found between the bulk data versus the myeloid-specific data that you can get from single-cell data. So in the bulk comparisons, we can see the expected pathways, such as upregulations in stem cell genes. But if you do single-cell studies, the myeloid cell-specific comparisons comparing the disease versus the normal control review single-cell studies to look at upregulation of specific pathways. For example, now we see this FLT3 pathway is being perturbed. This is going to be missed by bulk RNA-seq analysis. Now, why is this FLT3 important? Because these gene mutations is found to be present in about 30% or 1/3 of the AML patients. So overall, single-cell analysis allows you to dive deeper into looking at biological complexity, which is not possible if you're using bulk methods. So at 10x Genomics, we look at complexity in biology by using molecular barcodings. So you can maintain the identity of the single cell or the tissue spatial regions, but at the same time, you can process the sample in bulk using our end-to-end workflows process. So the key for the 10x technology is that we can barcode the targets efficiently and effectively, such a way that you still maintain the identity of the single cell or the tissue regions, along with additional capture features that you want to do along with the transcriptomes. So for example, today, I'm going to focus on our 10x Chromium single cell platforms. And the 10x Chromium single cell gene expression platforms has been highlighted both in the Nature Methods, in their Methods of the Year, and also in the Scientist Top 10 Innovations for the last couple of years. Today, due to the time limitations, I'm going to focus on our award-winning Chromium single-cell 3' gene expression solutions, which has generated over 2,000 publications, not including preprints. Now, note that if you're interested in profiling the T-cell and B-cell receptor along with the transcriptome, or to look at T-cell receptor or the peptide MHC antigen interactions, we also have the single-cell immune profiling solutions. If you're interested to look at the epigenomes, we have the single-cell ATAC-seq solutions. If you want to profile the epigenomes and the transcriptome simultaneously from each of the nucleus, we also have the single-cell multiome solutions. Even though today I do not have time to talk about all these other single-cell applications, if you're interested, please contact us at any time. So the Chromium 3' gene expression solutions with the feature barcode technology allows you to do both the whole and also the targeted gene expressions analysis at the single cell or the single nucleus levels. So we have the optional feature barcode technologies. So along with the transcriptomes, these provide simultaneous assessments of perturbations if you're doing CRISPR screening to do knock-in, knock-out of genes. You can also perform quantitative analysis of protein abundance, as in the case of cell surface protein profilings from intact cells. One thing I want to mention is that the gene expression solution from 10x is very easy to use. It is very robust, and then it is compatible with demonstrated protocols for a variety of sample types, including cell lines, primary cells, dissociated tissues. Our technology is compatible with whole cells and nuclei. Another thing to mention is key to 10x technology is that we also contains turnkey analysis and visualization software tools that you can download from the website to analyze and look at the 10x data. This slide gives you an overview of the 10x Chromium single-cell workflow. You're going to start with dissociating your samples into single-cell or nuclear suspensions. And then you make these single-cell droplets using the Chromium controller and the single-cell reagents. Our reagents and consumables contain the gel beads that come with the reagent kits, the microfluidic chips for you to make the single-cell containing droplets, and also the library construction reagents. The workflow is going to generate libraries that are ready for various Illumina short-read sequencing instruments. After you sequence the library, and then you analyze the NGS data using the 10x Cell Ranger pipelines, And then you can also look at the results using the 10x Loop Cell browser. You can download both of these software analysis and visualization tools from the 10x website. So how does 10x technology works? The workflows for you to make the single cell library starts by combining these bar-coded sequences with high resolution partitioning of these input cells and nuclei into each of these individual droplets of emotions inside the microfluidic chips using the Chromium controller. So during this workflow, the single cell or the single nucleus suspensions are going to be combined with the reagent mix that you loaded into the Chromium chips, along with these 10x gel beads. And you also add oil that come with the reagent kits. So the gel beads are 10x primer delivery systems, and they're indicated by these color circles on these diagrams. After you load all these reagents and cells into the chips, you then place chips into the Chromium controller, and the controller is going to make these droplets that we call the GEMs, stand for gel beads in emotions. So on this slide, you're looking at the single cell 3' gene expression kit. These chips enable partitionings of the individual input cells and nucleus, along with the 10x bar-coded gel beads into the droplets. Up to 8 of these channels can be run in parallel at one time on a single-use chip. If you're using our standard 3' gene expression kit, each of these channels can recover between 500 to 10,000 cells per channel, and you control how many cells you want to recover. At 10x, we have tested cells up to 30 microns in diameters. In theory, however, you can recover cells up to 50 microns in diameter, which is the width of the microfluidic channels. The systems also provide the flexibility for you to run less than 8 samples per chip at one time, as long as any of the unused wells, you're going to fill them with 50% glycerol. The chips cannot be used more than once, regardless of whether samples or glycerols were loaded into each of the channels. So the partitioning process using the Chromium controller only takes about 18 minutes. And during these partitionings, about up to 85% of the input cells and nuclei can be recovered and captured. So this means that if you load approximately 16,000 cells, and then you can recover the 10,000 cells per channel using the standard kit. Now, most of the occupied droplets of the GEMs will contain a single cell, but there is a statistical probability that some of the droplets of GEMs may contain more than one cell. So the expected fractions of the droplets with more than one cell actually increases with more cells that you recover. If you're looking at the standard kits, for every 1,000 cells that you recover, you only get a very low multiplet rate of about 0.8%. And this is the data that we normally filter out for single cell data analysis. So during the Chromium run, the cells of the nuclei and the gel beads are combined into these one solution streams in this channel within the chips. So these solutions flows through the channel and pass this interface of the recovery well, and also pass the channels containing the cells and the gel beads, and droplets are formed at this interface. So once these are formed, these GEMs are collected in the recovery well. So these live action videos on the right shows the formations of the GEMs inside the chips in the Chromium instruments. So you can see these gel beads are being fed into the main channels in the chip. On this slide, you're looking at a GEM. Most of the GEMs will contain one 10x gel beads. A subset of them will contain a captured single cell or nucleus. In this diagram, you're actually looking at an encapsulated single T cells. So after these GEMs are formed, the gel beads is going to dissolve, release the primers into the system, and the cells will lyse, releasing the nucleic acids into the solutions. So in our 10x gene expression solutions, within the GEMs, when you incubate these droplets on the thermocyclers, reverse transcription happens in the RNA-seq assay. It's going to convert the poly-A containing RNA and also the optional features of interest in the 10x bar-coded cDNA and also the 10x bar-coded feature DNA, respectively. So gel beads are the 10x primer delivery systems. Showing on this slide is a schematic representations of the 10x single cell 3' gene expression gel beads. it contains 3 different oligotypes. This first one is used to capture all the poly A containing RNA molecules. This includes all your mRNAs. The components of these oligos contain a partial read-one sequence, a 10x barcodes. This is used to identify each of the cells or nucleus in which the transcripts or the feature is found. So all the primers in one gel beads, they all contain the exact same 10x barcode sequence. But each of the gel beads, each will have a different 10x barcode sequence. This enables bioinformatics analysis for us to identify each of the unique molecules that come from each of the cells of the nucleus. The next component is this UMI molecule. This stands for Unique Molecular Identifier. This is used to identify and count the origin of molecule that are present for each of the genes in each of the cell. So essentially, the UMI is used to tag each unique cDNA that is synthesized from each of the poly A containing RNA molecules. And all the primers in one gel beads, they contain a random number of 12 base pair of UMI. The last component is this poly DT tail. So these will bind to the poly A tails in the transcripts, enable you to capture all the poly A containing RNA molecules. In addition to these poly DT containing primers, we also have these 2 additional oligos in the gel beads. These contain this capture sequence 1 or capture sequence 2. These are used to capture any additional sequence that you want to profile along with the transcript terms. So what you do is that you can design an oligos that attach to those analytes that contain a complementary capture sequences. So after barcoding, and you're going to amplify the cDNA, and then you perform the final library construction steps in bulk, collecting all the cells in one tube. And then you make into a final NGS library that you can sequence. So the library preps involve cleanup, enzymatic fragmentations. For the 3' gene expression assays, you retain the 3' N, followed by N repair, A tail ligations, and then you add in the sample index, and then to generate final Illumina compatible libraries that you can then sequence with the Illumina instruments. So for sequencing, if you are looking at whole transcriptome measurements, that is, you want to look at all the genes that are expressed, we recommend to sequence a minimum of 20,000 read pairs per cell for the whole transcriptome analysis. So note that other than whole transcriptome assays, you can also look at a subset of genes using our compatible targeted gene expression assays, which I'm going to talk about later. So the sequencing configuration is 28 bases for read one. These include the 16 bases of 10x barcode, 12 base pair of the UMI, and the 2 indexes will be 10 base pair each, and read 2 will be an insert, and we recommend a minimum of 90 bases for the cDNA insert. So after you sequence the libraries, you can then use the 10x Cell Ranger pipelines that you can download from the 10x website to analyze the gene expression or the gene expressions with the optional feature barcode libraries. So note that at 10x, we have a pipeline called the MakeFastQ. So this is a 10x barcode-aware demultiplexing that you can feed in your BCL files and then to proceed all the way to the analysis. You also have the options of feeding in the FastQ file as the input. So we have the Cell Ranger count, which is the main pipelines in a 10x system. So this provides the read level analysis of the single cell libraries, and is going to produce the feature barcode matrix. So the results for the analysis is a digital single cell, single nucleus gene expression profiles with the optional features, with the optional informations on additional features of interest, such as CRISPR perturbations and cell surface protein expressions that you can profile along with the gene expression assays. In addition, the Cell Ranger pipeline also produces C-loop files that you can use and visualize using loop browsers, and you can use it to perform some simple analysis, such as differential gene expressions analysis, and then you can look at differences between these different clusters of cells. Now, other than whole transcriptome readout, you can also perform targeted gene expression assays, and this is compatible with our 3' gene expression solutions. So the targeted workflows use the final sequencing library as the input for target capture by Bates. Our targeted libraries can also be sequenced on a variety of Illumina sequencing platforms, and you can also analyze the data using the software tools that you can download for 10x website to perform targeted analysis. If you're studying humans, we offer 4 pre-designed human panels: a human pan-cancer panels, a human immunology panels, a human gene signature panels that you can use to profile a wide variety of important signaling pathway, diseases, and drug targets. We also have a human neuroscience panels. All the gene lists for our panels are listed on the 10x website. In addition, we also have a custom panel designers. With this, you can create your favorite designs. You can add up to 200 genes to any of the pre-designed human panels, or you can do a fully custom human panels that contain 10 to 1,500 human genes. In addition, you can also add up to 10 non-human exogenous sequences to any of the panels. So if you want to do the targeted gene expression assays, so the library structures and the required sequencing configurations in terms of the read lengths are identical to the 3' whole transcriptome libraries, but the recommended depths different. So the recommended sequencing depths for the targeted panels depends on the panel size and contents that you use. For any of the human pre-designed panels, we recommend a minimum sequencing depth of about 2,000 read pairs per cell, and further optimization may be needed depending on your cell type, the panel size, and also your favorite panel compositions. Now note that in addition to the standard kit that I talked about, where you can recover between 500 to 10,000 cells or nuclei per channel, we also have the 3' low throughput gene expression kit. This kit is specifically designed for you to conduct smaller pilot experiments before you want to scale up. For example, if you're optimizing sample prep for new tissue types that you want to study, you want to generate some proof of concept data, you want to optimize some experimental conditions, these low throughput 3' gene expression kits can be a cost-effective way for you to start and run some of these small scale experiments. So with this low throughput kit, you can recover between 100 to 1,000 cells per channel. And then these products work with the same wide variety of sample types that you can do with the standard kit. The cell recovery rate for the low throughput kits is about 30%, with a multiplet rates of about 8% per 1,000 recovered cells and nuclei. So next, let's look at examples on the single cell data. Here you're looking at the primary cell population PBMCs that we prepare from blood, and there are a wide variety of cell types in PBMCs, including both the innate and adaptive immunity cells. Using the 10x Chromium single cell assays and through marker gene analysis, you can discover major populations that form in these different clusters. For example, here you're looking at t-SNE projections of the whole transcriptome expression profiles from about 68,000 unsorted human PBMC. Each of these dots is a cell. So these t-SNEs actually look at the expressions and all the genes, and it positions the cell with the same gene expression profiles together. Cells that are further apart means that they have different transcriptional profiles. And the colors indicates the clusters identified based on marker gene expressions. So overall, this reviews the high-level structures in an unbiased t-SNE projections that reflects the known cell types present in PBMCs. Now, one thing I want to highlight is this CD34 positive myeloid progenitor cells, which is only 0.3% in abundance. You will never be able to identify these populations in bulk studies because the signals are masked and diluted out by all these other signals. If you're using flow cytometry to identify cell types, you will not be able to identify all the cells, especially those without markers that you can use for flow sorting. Now, as single cell biology can be used to look at the complexity of a variety of biological systems, a great use for this is the global initiative, the Human Cell Atlas projects. So the Human Cell Atlas project is an international collaboration that aims to look at all the human cell types, define them in terms of the molecular profiles, such as gene expressions, and to connect these informations with the classical cellular descriptions, such as locations and morphology. The aim is to create a comprehensive reference map of all the human cells as a basis to understand human health. So much like the Human Genome Projects that Dr. Riza just mentioned, that has fundamentally changed the way how we explore biologies, and this is more than a decade ago. This Human Cell Atlas and other initiative around the single-cell genomics, transcriptomics, and epigenomics it is again going to change the way how we explore biology, ultimately to influence how we treat diseases. And we are very happy to see that our Chromium single-cell solutions, including the 3' gene expression solutions I talk about, is being actively used in the Human Cell Atlas projects. At 10x, it is our missions to further these studies, to review the complexity of biologies through all the innovative solutions that we provide to you. With that, if you have any questions or you want to dive deeper into any of the topics or other topics I do not talk about today, please feel free to check out the resources we have online. We have webinars, scientific talks from various researchers in a lot of application segments, including oncology, immunology, infectious diseases, neuroscience, and others. We also have a lot of resources online at your disposals, and you can also contact our local support team, and then we can also provide assistance to you. With that, thank you, and I'll pass it back to Meta.
Yeah, thank you so much for your great presentation from Leo. Kita akan